Hello, everyone. We are starting our discussion of skeletal muscle physiology by looking at the structure of the skeletal muscle cells. There's a really important core concept in physiology that states that the structure of a cell really represents its function. And it's true in all cells, but I feel like this principle is especially well illustrated when we talk about the skeletal muscle cells. So that's why we're going to take this video really to focus on the structure or the anatomy of these skeletal muscle cells, because I think it lays a really good foundation to then understand how they work or what their function is. So here are the learning objectives, which I encourage you to come back to after the video to make sure that you got all of the important concepts. So as you know, this lecture and the next few lectures focus on skeletal muscle. So these are the muscle that people typically think of when we refer to muscle. They're the ones that are attached to your bones and that make your skeleton move, like your biceps, your quadriceps, your gluteus muscles, and so on. But these aren't the only types of muscle that we have in our body. We actually have three types of muscle. So apart from skeletal muscle, we also have cardiac muscle. And these are muscle cells that are found only in your heart, as the name says, and their function is to move blood through the circulation. We will discuss in detail the function of cardiac muscle in 1002 or 1012, so in the winter semester. And the third type of muscle is smooth muscle. And these are found lining your internal organs, like your stomach, your bladder, and your blood vessels. So when you think about it, all three of these muscle types generate motion and force. Your skeletal muscle, as we discussed, help move your skeleton or help move your body into space. Cardiac muscle cells will help move blood through the heart chambers first and then subsequently in the circulation. And finally, smooth muscle cells move substances to different internal organs. It helps to move food through our digestive system. It helps to move urine through our urinary tract. It helps to move blood through our blood vessels, for instance. So skeletal muscle are the focus of this video. There's a few things that differentiate them from the other two types of muscle. One of their characteristics is that they are under voluntary control. So if I ask you to contract your biceps right now, you likely are able to do that. If I ask you to raise your toes, you're also able to do that because these movements are controlled by skeletal muscle. That's in contrast to the other two types of muscles, which are not under voluntary control. If I ask you to make your heart beat stronger, you have no control over that. If I ask you to narrow your arteries, you can't do that either. So this voluntary control is a characteristic specific to the skeletal muscle cell. The other characteristics that's unique to those cells is that they are very, very large cells. They are actually the largest cells in our body. And that's because one skeletal muscle cell is actually made of tens or even sometimes hundreds of precursor cells in development. So what this means is that when you look at muscle cells under the microscopes, they don't have one nucleus like most other cells in our body. They can have many, many, many nuclei. And the final characteristic that I want to bring your attention to is the striation of skeletal muscle. And you'll see that skeletal muscle as well as cardiac muscle are called striated, while smooth muscles are called non-striated. And what this means is that when you look at these cells under a microscope, they have striation, which means that they look like they're striped. So if this is a cell, skeletal muscle cell might look a little bit like this, with stripes of dark band and light band next to one another. So now that we know that, let's try to match each muscle type with its appearance under a microscope. So we have one type of muscle here, the second, and the third. On the right-hand side, we have actual microscope images from these muscles, and on the left-hand side, you have the representative cartoon. So let's focus on the cartoons first. So going back to our striation concept, you can see that this image and that image have the stripes that we're talking about. You can see there's one dark band, one light band, one dark band, one light band, and so on and so forth. You can also see this pattern under the microscope, especially here you see the stripes really, really well. Here you see the stripes in the cartoon, and the stripes are here in the microscope, but they're not as obvious. So we know that this is a striated type of cell, and this is also a striated cell. So if we label the first one as A, this one is B, and this one is C, our B cell is a non-striated muscle, which means it is our smooth muscle cell. So now we have to differentiate between our skeletal muscle and our cardiac muscle. Let's try to look at one cell. So I'm going to highlight one cell here. This is one cell. And here, this is one cell. First of all, you can see a size difference. This one is bigger than that one. The second difference you can see is the number of nuclei within one cell. Here, we have one cell, one nucleus. The nuclei here are in blue. And here, within one cell, we actually have two nuclei, one here and one here. And as we just talked about, an actual skeletal muscle cell can have way more than two nuclei, 10, 50, even hundreds of nuclei. So based on that, our cell with many nuclei is our C, and that represents our skeletal muscle, while our striated cell with only one nucleus is our sample A, and that represents our cardiac muscle. Let's now look at function. So can we actually match each of the muscle types with its function? So which muscle type would be used to wave hello? The way to think about these questions is first to ask yourself whether waving hello is something you have voluntary control over or not. And obviously, in this case, the answer is yes. So you have voluntary control over waving, which means that this is done by skeletal muscle. Mixing food in our small intestine. We have no voluntary control over this. This does not relate to our heart. Therefore, this is done by smooth muscle or number 3. Your heart beating is done by your cardiac muscle. Airways constricting during an asthma attack. You have no voluntary control over this. The smooth muscles are responsible for that. So moving on to E, taking a deep breath in. That's actually an interesting one. So go back to the question. Do you have voluntary control over your breathing? If I ask you to stop breathing right now for a few seconds, you'd be able to do it. If I ask you to breathe really, really quickly, you can also do it, at least for a few seconds. So we do have voluntary control over our breathing muscles, and they are indeed skeletal muscle. Shivering, that's also an interesting one. So shivering is actually the contraction of skeletal muscle. And our body uses shivering, as you know, as a way to produce heat to bring our body temperature back up when we're cold. So our skeletal muscles are responsible for shivering. And next time that you shiver, we actually have some voluntary control over it. If we try really hard, we can stop shivering for a few seconds. And finally, increasing blood flow to specific organs. Let's say your brain needs more oxygen. What kind of muscle are responsible for increasing that blood flow? We actually have two answers here. First, we need our heart to increase its output. So your cardiac muscle will be involved in that. And second, we need the arteries that go towards the brain to be as wide as possible so that more blood can get through. And your smooth muscle will be responsible for this. So hopefully this little exercise helps you differentiate a bit more between the functions of the 3 different types of muscles. So let's now focus on skeletal muscle and its function. So I want to bring your attention to the cartoon first. So what we see here is an entire skeletal muscle. And that skeletal muscle has been cut. So here we're looking at inside of the muscle itself. And each of these cylinders are actually called muscle fascicles. So this is one muscle fascicle. And let's blow it up even more. Within the fascicle, you have all of these cylinders, which represents one muscle cell. So what we have here is one muscle cell, which is also called muscle fiber. So let's recap here. A skeletal muscle contains many muscle fascicles, and within each of these muscle fascicles, there are many muscle fibers. Now, muscle fibers and muscle cells mean exactly the same thing. So you can use one or the other and it means the same thing. So muscle fiber or muscle cell. Now, within the skeletal muscle, there's not only muscle cells. There's also other types of tissues and other types of cells. So there will be connective tissues that will hold these fascicles and hold these muscles together. And there are obviously nerves and blood vessels, which you can see here in blue and in red. So you can think of a skeletal muscle as a collection of muscle cells. Just like when we talked about neural physiology, a nerve is a collection of neurons. So now let's look at what is actually inside a muscle cell or a muscle fiber. So within the muscle fiber, we find sarcoplasm, sarcolemma, and sarcoplasmic reticulum. Now, we haven't encountered these specific words yet, but we've encountered the concepts. So as you can see here, sarcoplasm is just a different word to indicate cytoplasm. Sarcolemma is another word to indicate the plasma membrane of that muscle cell. And finally, the sarcoplasmic reticulum is another word for an endoplasmic reticulum. So you see that these three words all start with the same prefix, which is sarco. And that's because this prefix sarco means flesh and is used to refer to structures of the muscle cells specifically. So sarcoplasm is the cytoplasm of a muscle cell. The sarcolemma is the plasma membrane of a muscle cell. And the sarcoplasmic reticulum is the endoplasmic reticulum specific to the muscle cell. So now let's go even a little deeper inside one muscle cell. So we've already mentioned that one muscle cell contains many nuclei, the sarcolemma, as well as a sarcoplasm. So in the cartoon below here, this is a blow up of one muscle cell. So this is one muscle cell, and we've cut it open to see what's inside. So this is what's inside, and it's color coded. So what you see in blue here is the sarcoplasmic reticulum. The main function of the sarcoplasmic reticulum in muscle cell is for calcium storage. So inside the sarcoplasmic reticulum, there's really high concentration of calcium, and we'll see how that's important for muscle contraction. In purple here, we have mitochondria. And mitochondria are responsible for ATP production. So the more mitochondria a cell has, the greater its capacity to produce energy. So think about your skeletal muscle, which need energy to contract. It's really important to have lots of mitochondria in skeletal muscle, so we always have energy not only to walk and to run, but even just to hold our posture, to breathe, and to respond to emergency. We don't see it on the cartoon, but muscle cells also have a lot of glycogen. The role of glycogen is to provide energy. So as you may remember, glycogen can be converted to ATP. And finally, in the pinkish red here, we have myofibrils. And myofibrils are responsible for the contraction of skeletal muscle. We'll come back to those in a few seconds. Now I want to bring your attention to the sarcolemma. Remember, the sarcolemma is the plasma membrane of the skeletal muscle. When we think about the plasma membrane of a cell, we often think of it this really nice round shape. It's not the case in skeletal muscle. In skeletal muscle, the plasma membrane has those dips. I'm going to try to highlight them here. So plasma membrane here, and then it dips here. And then plasma membrane, and it dips here. And so on and so forth. So all of these dips are called T-tubules. T actually stands for transverse tubules. I'm going to write it down here. So the T stands for transverse. And those T-tubules are everything that you can see in the gold color on the cartoon here. There's a key concept here, and it's illustrated right here. So you can see that the T-tubule, which is in gold here, is very, very close to the sarcoplasmic reticulum, which is in blue here, on either side. There is actually a physical link between the T-tubules and the sarcoplasmic reticulum. And once again, Remember that structure matches function. So it's not randomly that there is a physical link between the T tubules and the sarcoplasmic reticulum. There is a very important function for that when we'll talk about the contraction process. Now we're going back to the myofibrils. We said the myofibrils are responsible for the contraction of muscle. And they do that because they contain sarcomeres, which are the contractile structures of the muscle cell. And we see a sarcomere right here. So a sarcomere starts from here to here. Okay, so that would be one sarcomere. And basically, a myofibril is a long line of sarcomeres, one next to the other, next to the other, next to the other. So our next slide, we're really zooming in to what is inside a sarcomere. So at the top here, we have a section of a myofibril. For instance, here we have two sarcomeres in a row, one from here to here, and then one from here to there. But what's really important in this slide is the structure of the sarcomere. So the sarcomere has many different proteins that will participate in skeletal muscle contraction. The two most important proteins that are directly involved in contractions are actin and myosin. And once again, it's all color-coded. So the actin forms the thin filament, which we see in red. And the myosin forms what we call the thick filament, which we see in purple. So the sarcomere starts here at what we call the Z disc and ends at the other Z disc, which is right here. So the actin is on the outside of the sarcomere, and the myosin is on the inside of the sarcomere. Now, in order to keep those positions, these proteins must be anchored by other proteins. And that's the role of the accessory proteins. So titin here is a protein seen in green. It links the myosin, in purple here, to the Z disc, basically making sure that the myosin strand doesn't move and stays in its place in the sarcomere. So we can say that titin anchors myosin. Conversely, nebulin anchors actin. So nebulin anchors actin. So it makes sure that the thin filament in red here stays there. So we don't actually see nebulin in this cartoon, but we'll see it in our next slide. And finally, the sarcomere also has two regulatory proteins, troponin and tropomyosin, which we'll see next. So let's look specifically at the thick filament and then the thin filament. Remember that the thick filament is formed by myosin proteins. And the myosin molecules contain a tail, a hinge region here, which is going to allow those heads to move position. So the head can basically swivel in this direction and that direction, depending on the position of the hinge region. What's really important here is that the head of the myosin have binding sites for two important proteins. So they have a binding site for actin, and they have a binding site for ATP or ADP. So the myosin head is able, through its ATPase activity, to take the ATP, break it down into ADP, and then use that energy to power skeletal muscle contraction. Now let's look at the thin filament, which is formed by actin. So actin is actually a globular protein. So think about it as a sphere or a ball. And it's represented here with all those red balls. So each of those is one actin molecule, but they all interact together to form this filament. Now, as you can see, in the thin filament, there's more proteins than just actin. First of all, we can see nebulin in that thin strip of blue here, which, as we said, anchors actin, makes or make sure that the thin filament stays in place. There's two other proteins that associate with the thin filament. The first one is tropomyosin, which is represented in that turquoise here. And the second one is troponin, which is represented in the brown. So one of the key characteristics here is that actin actually has binding sites for myosin. So I'm making those up here in blue. Label those myosin binding site. So hopefully you can see here that myosin can bind to actin because it has a binding site, and actin can bind to myosin because it has a myosin binding site. So during contraction, actin and myosin actually physically interact with one another, or they bind to one another. And when that happens, we call these cross bridges. Now, one of the problems is that those myosin binding sites here are not always available. In fact, sometimes they are hidden by tropomyosin. So in order to produce a really powerful contraction, the cell needs to move tropomyosin out of the way so that the myosin binding sites become active. So now let's recap some of the important structures that we talked about. The first one is the sarcomere. So a sarcomere goes from one Z disc to another Z disc. And the two main proteins that are important here are actin and myosin. The actin forms a thin filament, which is located on the outside of the sarcomere. So this is actin that forms a thin filament. The actin also forms the light part of the sarcomere. Remember when we talked about the striation of the skeletal muscle? This striation actually represents actin and myosin. So when you look at the sarcomeres and there's only actin, this shows up under the microscope as a light color. Conversely, if you look at myosin, this shows up as a dark color. So that's why we have this pattern or light, dark, light, dark, light, dark in our skeletal muscle cell. So that's actin. In the middle of the sarcomere, we have myosin. And all of the tails of the myosin are in the middle. And then you have the head of the myosin towards the outside of the thick filament. So these are the heads of the myosin. That goes in both directions. So once again, this is your myosin, which forms your thick filament. And shows up under the microscope as a dark bed. Then we have our accessory proteins. So titin actually is the largest protein known in the human body right now. It's the protein that's going to anchor the myosin. So one molecule of titin, which I just drew here, that's one molecule, can actually have up to 25,000 amino acids. Okay, this is a giant protein. And obviously it's present on both sides. And finally, it's hard to draw, but we have nebulin. I'm going to draw in blue here. That's our accessory protein that helps keep our actin in place or thin filament in place. So hopefully this helps clarify the important structures within the sarcomere. For our last slide, we're looking at the organization of the thin filament. And I haven't mentioned it for a previous slide, but I really do encourage you guys to draw with me as I draw this. It's a really good way to solidify that knowledge if you are able to draw those structures first with our support and then by yourself. So take a pen, take a paper, and follow along. All right, so as we said, our thin filament is made of a chain of actin molecules. So each of those circles represent one actin molecule, which together make up the thin filament. So a true thin filament would have many strands of actins that would weave in and out. But for simplicity's sake, I'm just drawing the one strand of actin right now. And we know that the actin has myosin binding sites, which I'm drawing in blue here. So each actin molecule has a myosin binding site. Now, when a muscle is not contracting, tropomyosin is actually located on top of the myosin binding sites. So it basically hides those myosin binding sites. So in green here, what I've drawn is tropomyosin. And the key concept is that it hides the myosin binding sites. So during contraction, the cell will need to move this tropomyosin out of the way so that actin and myosin can interact together. To do that, it uses the protein's troponin, which I'm drawing here in purple. One of the key characteristics of troponin is that it has a calcium binding site. In other words, when calcium concentration within the cell goes up, it can actually bind to troponin. And that's going to be the signal to move the tropomyosin. So here's a recap of our thin filament and how actin, tropomyosin, and troponin are organized related to one another. So I hope that helps clarify the structure of a skeletal muscle cell, and that lays a foundation for all of the skeletal muscle functions that we're talking about next.